Thank you, everyone. I'm Huan Zhang, and I'm a PhD student from the University of Tokyo. And today my topic is evolution in the subfamilies of all human endogenous retroviruses. Okay, so what is endogenous retroviruses? Actually, in our human genome, there are about 80% of the human genome comes from retroviruses, including some genes and a regulatory element. And the endogenous retroviruses actually are ancient retroviral sequences found in the genomes of mammals, including human genomes. And they are derived from retroviruses that historically infect germ cells, becoming integrated into the host genome. And the types of them are classified based on non-terminal repeat. That's shown here. Long-terminal repeats, and mostly they are harmless irreversible rules in gene regulation and genome evolution. They are linked to certain diseases like cancer and autoimmune disorders. So our topic focuses on two subfamilies. They are MLT2A2 and MLT2B3. Actually, they belong to MLT2 family. So MLT2 and minor, they are family of ERV3, and ERV3 is a type of ERV and a kind of transposable element. So why do we need to reclassify MLT2A2 and MLT2B3? As we can see, so here we have to introduce a concept of hybrid transposable element. So here we have two transposable elements, A and B. So the LTR, right LTR of A, recombined of the left LTR of B, and then the hybrid transposable element formed. And this kind of element is hard to be detected by most of the tools. And that's why in this research we use loss. Loss is more accurate because it can match each part of the sequence to a different part of the genome through loss split. Let's see the example here. So on the above is the loss result. So the loss result tells us actually this element consists of two parts, MLT1B and MLT1C. But the repeat marker, the most used annotation tool, told us it's just one element here. So the repeat marker cannot tell the element when the element is hybrid. So that's why we use loss here to detect the transposable element. And then we extracted those hybrid elements. And this figure shows the hybrid elements occurred more than 100 times in the human genome. As we can see, hybrid elements here occurred more than 2,000 times in the human genome. And we named it as ABA because it consists of MLT2A2, MLT2B3, and MLT2A2 again. And this is the figure show what it's like. So in the human genome, as we can see, the loss tells us it consists of three parts, like I said. But the repeat marker, two versions of repeat marker give us two different answers because it cannot tell what actually it is. So the first one version of it tells us actually is MLT2B3, but another version of it tells us it's MLT2A2. So repeat marker just it can tell actually this element similar to these two elements, but cannot tell what actually it is. So this is the evolution tree that it tells us actually this. So we named this hybrid element as ABA, and as we can see from the tree, this ABA is actually distinct from any other subfamilies in the MLT2 family. So we can see actually these hybrid elements can be seen as a new subfamily. So that's why we need to reclassify them. But the next question is how to reclassify. So currently we are here. We have used the loss to collect instances of MLT2A2, MLT2B3, and the newly constructed ABA. Okay, we have 3 groups here. So next step, we need to use the repeat modeler to reclassify them and to construct consensus sequences. What is a consensus sequence? It's like the one that can represent all of the instances. So in each subfamily, we have many instances, and we have to select one to represent all of these instances. Okay, so here is an example. So we use repeat modeler. The repeat modeler is mostly based on the method multiple sequence alignment. And further extractions will be made if potential subfamilies were found. What does it mean? So this figure shows the consensus sequence. The top one is consensus sequence aligned to many instances. And this figure shows that some of the instances will match to the consensus sequence here, 4 match aligned. And some of the instances here, there seems like some other structure here, and only some parts, 300 pp of them, aligned to the consensus sequence, and another 100 pp aligned to another part of the consensus sequence, and in the middle there is a gap. So these instances, maybe a new subfamily needs to be separated from the current database. Okay. So once we found the potential subfamilies, the next step, we have to check them. We have to check if they are really subfamilies need to be separated. So there are 2 steps to do. The second step is easy to understand, determine their abundance in the genome. That means if there are just 2 or 3 instances in the human genome, then there's no need to reclassify them. And the first step confirms the presence of fixed pattern in the sequences. That means here we can see, like on the left, there is a subgroup that has different sequences at their 3' end. And here, this subgroup has a spatial, they end with ACCCTAA, but other instances end with ACCDGA and something on. So this subgroup is different from other sequences. So this is very spatial. And next, we will check other details like divergence. So basically, the divergence should be keep the same, like this about 10, and some details like how the instances align to the consensus. And then we got the results like this. So the original MLT2B3 instances, we found a subgroup can be further separated as SAB-B3 based on the multiple sequence align result. And also the ABA, the ABA is we newly found subfamily, but we can also further separate another subfamily from them. That is, we named it as sub-ABA. So this is the details of the MLT2B3 compared to the SAB-B3 instances. Like we can see, the horizontal is the consensus sequence of MLT2B3 and ABA, while the vertical is instances of SAB-B3 and sub-ABA instances. So speaking of the MLT2B3 consensus sequence with SAB-B3 instances, they are different from about 250 pp here, and ABA is different with SAB-ABA at about 300 pp. So actually they are different. So originally we have just 2 in the database, current database, there are just 2, MLT2A2 and MLT2B3. And we found actually there is a subfamily can be separated, that is ABA. And inside ABA, we found a sub-ABA. And inside MLT2B3, we find sub-B3. So finally, we got 5 subfamilies here. And this figure shows the length of each subfamily. Okay, so this is inner evolution tree. So this tree is near perfect except for the ABA. So ABA seems to appear twice in the tree. So here is a speculation, maybe ABA have some other unknown evolution, or ABA. Inside the ABA, we can separate another subfamily. We don't know. That is an interesting point. Maybe we can check later. And next is the unrooted tree. So on the left, the tree is just constructed by the newly constructed 5 new subfamilies. It's a beautiful tree. And on the right, we add some outgroups into it, like some MLT1 elements and some other MLT2 elements in the MLT2 family. And as we can see in the tree, the blue one is the ABA. So ABA is like the center of other sequences. It has connections to some other subfamilies. So we can guess maybe ABA is an ancestral sequences of some other subfamilies. And then we project this newly constructed sequences in the human genome to other genomes, and we calculated the number of each genome in different genomes. So as we can see, the MLT2B3 may be the oldest one because it's generated from the Bush baby period, while others start from the Tertiary period. And we did the function analysis based on a previous study. So this study, the researchers found that MLT2A elements can act as promoters, and 81 of them are MLT2A2 elements, and one MLT2A2 element serves as a novel promoters of protein coding gene, GALNT13. However, based on our results, this only one MLT2A2 elements is ABA. So it's ABA that acts as a promoter. And also, among 81 MLT2A2 elements reported to be expressed in the 4 or 8 cell blastomeras, there are actually 25 ABA for MLT2A1, 43 MLT2A2, and 9 sub-ABA elements. And 2 elements with TPM more than 50 were sub-ABA and MLT2A2. So TPA means mean expression in embryo. That means although also our newly found subfamilies actually play an important role in the human genome, however, they're just been ignored based on the current Ronnie annotation or reclassification. So here's the conclusion. So our study revealed new subfamilies of MLT2 LTR elements and established new consistent sequences. And our results suggest that although transposable elements in the human genome have well started, there's still much room for research. And we mainly focused on MLT2A2 elements. However, we believe that that reclassifying transposable elements is a very rewarding and lengthy process. Thank you very much. That's all.
Thank you, Huan.
Yeah.
Any questions from the audience? No questions? Actually, my question is, where is the deep learning? Because you're parked here. So
Yeah.
maybe that's for the committee. Okay, so are you sure? No questions? Yeah.
So what is your standard to classify these subfamilies?
Okay, the standard.
Because according to the phylogeny, so the ABA is split into 2 parts. It's very fast.
Yeah. So as a first step, we found the ABA is based on the last So laws tell us that ABA is distinct from other any subfamilies. And then we do the multiple sequence alignment based on the difference of this subfamily of other subfamilies. So the standard is just the similarity within these subfamilies.
Yeah, so you don't consider the evolutionary relationship?
Yeah, we consider the evolution, but based on the sequence, like similarity, we have classified them into different subfamilies, and the evolution just based on the evolution tree or some other things.